Okay, so let me introduce to the actual system which we called molecular printing system, which you can see here. We also call it as a nano eNabler. So let me introduce this. So starting from the bottom part here, you can see that this is called substrate holder. So we place our targeted substrate on which the printing is intended to be done here after proper cleaning. So here, as you can see, I am going to put a clean silicon wafer, for example, over the substrate holder, and then we can properly insert the substrate holder into the MPS. After that, the second part is called SPT holder, and this is the part where we stick our SPT, and after that we dropcast the ink over the SPT. So I will show you how we place the SPT on SPT holder. Now we will dropcast ink here, AGNP ink on the SPT reservoir with the help of a micro pipette. So we saw that when we dropcasted ink over the SPT without treating the SPT in the UV ozone, we saw that the ink was not spreading well on the surface of the SPT. So to make the ink properly spread on the surface of the SPT, basically the surface of the SPT is silicon, so we need to do UV ozone treatment to make the surface hydrophilic to the ink. So in this section I will tell you how we do that. So this is the UV ozone pro cleaner which is used to treat the surface of SPT as well as the substrate in order to make the surface hydrophilic with respect to the ink, to attain the proper flow of the ink needed for controlled and desired printing. So here we can set the time, as you can see, from 10 to 60 minutes depending on the substrate and the ink, so what kind of interaction we want. So usually before initiating any printing experiments, we first of all clean the SPT as well as the substrate properly, and then just before the printing experiments we need to treat the SPT in this UV ozone cleaner. So we have to first open this and we put our SPT, a cleaned SPT, in the cleaner system. Simultaneously we can put our substrate also, and then we close the lid. After that we set the timer for how much time we need the UV ozone treatment. So here we are setting the time for say for example 10 minutes, and then we on the system. So the red signal means that the UV treatment has started. Also we need, this is a very important step, we need to blow the air on so that whatever the ozone is coming, to move that ozone air outside. So now we can see that the red light and the UV red light had turned off. So now it means that UV treatment process has been completed and we can take out our SPT and the substrate. But as I mentioned earlier also, it is very important to keep this blower on throughout the UV treatment process. It is so because when we open the lid, there can be residual ozone. So we want, to avoid our direct exposure to ozone, we need to keep this blower on. So slowly we can open the lid and wait for some 30 seconds, so that if there is any ozone present there, that can go out and we are not exposed directly to the ozone. So now we can bring out our SPT and the substrate, and then we can close the lid. After the UV treatment, we will check the wetting behavior of the ink. In other words, you can say we can check the hydrophilic or hydrophobic nature of the SPT surface after the UV treatment. So we will broadcast the AGNP ink again on the SPT treated with UV ozone. And now you can see here that we have been able to attain a proper spreading of the AGNP ink over the SPT surface. So now this condition is very important to attain a proper flow of the ink through the micro channels of the micro cantilever of the SPT. So one point here I want to mention is that to attain a desired flow of the ink, the interaction of the ink and the substrate is quite important. If a substrate is highly hydrophobic, then the ink molecules will not spread over the substrate. They will remain stuck to the SPT microcantilever tip. In the other case, if a substrate surface is highly hydrophilic, then the ink will spread very fast and it will have a high wetting parameter. In that case, it will be very difficult to control the flow of the ink and to control the dimension of the printed patterns. So that's why optimal balance between the hydrophilic, super hydrophilic, you can say, or super hydrophobic nature of the substrate is very important. So we need a balanced response of the substrate with respect to the ink, so that a desired structures can be printed with a proper flow of the ink. So now we will insert the ink loaded SPT reservoir inside the print chamber. The MPS system consists of a microscope whose objective directions can be controlled using these two knobs. Since the working principle of MPS is very much similar to AFM, we also have a laser source and correspondingly a detector for precise printing of smaller and final particles. Moreover, to control the humidity inside the print chamber, we have inbuilt humidifier which is placed here. So once we have properly placed the substrate and the ink loaded SPT inside the print chamber, so once the printing chamber is closed, we initiate our printing experiment. So in this section, we will show you the MPS user interface. We call it a nanoware. In the left hand side of this interface, we have controls for the X, Y and J movement of the SPT. Also we have the controls for objective. In the right hand side of this interface, you can see the cantilever of the SPT, which is right now in the focus, and the unfocused area is the substrate. In the bottom part of this interface, we have humidity control section. This section shows the current humidity and this section shows the target humidity. Right now it is 50. If you want to change this humidity, say for example 60, we put the value here and according to the target humidity, the printing chamber tries to attain that value with the help of inbuilt humidifier. In the next section, we will discuss the different parts of the nano air interface. So now, once we have loaded the ink over the SPT and placed the SPT on the SPT holder and then SPT holder in the chamber, so using this nano air interface, user interface, we can now clearly see the presence of the ink. So now the ink particles, in short, we can say that ink has flowed down to the through the micro channels of the cantilever till the tip of the micro cantilever, and this is ready to be printed. Also, one point I would like to mention is that using this interface, we can measure the dimensions in the micron regime. So here you can see that the length of the SPT is roughly 200 micron, and if I zoom in more, here you can clearly see that the width of the micro cantilever, the total width of the micro cantilever of the SPT is near 60. So that's why we call it 60 R or S. And we can see that the micro channels are of width 10 micron here. So we have these two channels through which the ink is being flown. So ink is able to flow through these channels and is ready to be printed.
MPS is used for direct deposition of materials on targeted locations over a substrate. Using this printing system, we can print nucleic acids, proteins, lipids, DNA, and also non-biological materials like metallic inks, polymers, graphene, etc. on diverse nature of substrates like silicon, glass, PVA, PMMA, and also flexible substrates like PET. The heart of this printing system is called surface patterning tool, a schematic of which is shown here. It consists of a supporting substrate, and the print head is microcantilever, which is made of thermally grown SiO2. It consists of micro channels, and the material to be printed is drop casted near reservoir in the form of ink. The ink particles flow through this micro channel of the cantilever and arrive at the opening gap or micro cantilever tip, and when this tip touches the substrate, the ink is deployed and the printing is performed.